Hi, I'm Tamina Lebek. I'm a fourth year PhD student in the labs of Sally Lowell and Alistair Elphick, the IR at the University of Edinburgh. I'm really interested in cell communication, especially in the context of development. So how do cells coordinate these complex decisions that they have to go through to build a complex organism? And I'm really interested in if a cell decides to differentiate, is that communicated to their neighbors, to its neighbors, and if so, how? I wanted to see if there is a way where we can do neighbor labeling with a simple setup so we can implement it in many different cell types in a straightforward way. So we call the system PUFFIN, and this stands for Positive Ultra-Bright Fluorescent Fusion for Identifying Neighbours. So we have a super positive fluorescent protein that does the neighbour labeling, and that allows us to identify cells of interest, their neighbours, and their non-neighbours. My idea was to have cells secreting a fluorescent protein rather than a lot of systems are based on receptors or on a genomic switch in the receiving cells. But here the idea was to have a secreting cells, and that would be your cell of interest. And that sends out a fluorescent protein that can be taken up by any cell that is nearby. And we do this using a super positively charged version of GFP, and as I mentioned in the beginning, this is not really bright enough on its own, so we also use an amplifier, so that can be either a very bright fluorescent protein or, for example, the halo tag, where you can have the label in any color that you like. And then through that highly positive charge, it interacts with the negatively charged cell membrane of cells nearby, and all cell membranes are negatively charged, so it's a universal process of uptake, because if you have enough of that on the cell membrane, that then triggers endocytosis and you can detect the label in the nearby cells. And then we also need a second fluorescent protein in a different color that just remains inside the secretors, so we can identify the labeling cells from the labeled cells. But because then of these different fluorescent profiles you have, you can, for example, use microscopy or pull out the different populations using flow cytometry. And then if you can isolate these three populations, secretors, your cells of interest, the neighbors, and the non-neighbors, you can do functional studies or gene expression analysis on those cells that you sorted with flow cytometry. One of the main advantages I personally like about the system is our modular cloning strategy, so that you can easily exchange parts. So for example, you can also combine this with a gene of interest or a tissue-specific promoter. And the great thing is that all of it is on one single plasmid. And we kind of want to build like a puffin zoo, so where we try puffin in different model organisms, and we've already have some really nice evidence from chicken and mouse. We're now working with collaborators for zebrafish and flies, so Drosophila, and hopefully soon we have the xenoposter frog as well.